Thanks, everybody. It's an honor to be here and present to you. And just before we start, I will confess that Basil was I was the person that Basil spoke to last night. So I took his advice. I don't know what the bill is going to be like for this, but I thought that was a very good presentation. So I'm Neil McDonnell, CEO of Rock Therapeutics. And before I talk about, so we're a virtual pharmaceutical company working on a drug for NASH, as Jen said. Before I go into the company and what we're working on, I think a key part of this company is the team that we've constructed. And basically what we have is a team of 5 people that really started the company, 3 of us founders and a couple of others. All of us have many decades of experience in drug development in the pharmaceutical industry and biotech. And if you look here on our backgrounds, there's kind of this common thread of red, Takeda. That happened to be 4 of us happened to be working together at Takeda. This was kind of the dream team. And when Takeda got out of cardiovascular metabolic, where we were working, this was a place where we coalesced and decided, well, let's form a company based on one of the targets that Takeda was working on. Nothing I'm showing you today has anything to do with the work that was being done there, but we liked the drug target, and that's why we decided to form this company. So NASH, the mystery disease NASH. Everybody knows about cirrhosis from alcohol, et cetera. It's only been appreciated fairly recently that you can get that same endpoint just from living the way we all live, eating the way we do, not exercising. So all the things that contribute to metabolic syndrome result in non-alcoholic fatty liver disease. And then over time, that progresses to NASH, and NASH means you've got inflammation. And eventually you develop fibrosis and eventual cirrhosis. So the reason not many people have heard about it is if you go back and you do a lit search. There were 6 articles on NASH in 2000. There were 1,600 last year. So this is a disease that really has only been appreciated fairly recently, and there are no drugs to treat it. So it is a very hot area right now of interest for drug development. So the target that our drug works on is something called apoptosis signal regulating kinase, or ASK1. And what ASK1 does is it signals. It is something that's involved in helping the body deal with oxidative stress. So when a cell is under a lot of oxidative stress, that activates ASK1, and it signals down a couple of pro-inflammatory and pro-fibrotic pathways called the p38 MAP kinase pathway and the JNK pathway. And signaling down those pathways is what causes the inflammation, the apoptosis, and the fibrosis that constitute NASH. So the part of the reason that we were very interested in putting our ASK1 inhibitor into NASH is based on some data that Gilead, the biotech company here in the Bay Area, they have an ASK1 inhibitor, and we were watching them when we were at Takeda, and they showed the best anti-fibrotic response that's ever been shown in a clinical trial in NASH, in a phase 2 trial. And so this is some data from that trial, and this was really the data that got everybody excited. So their drug is called selonsertib, and they gave it at a couple of different doses, and then there was a control group. And this was in patients that had fairly advanced fibrosis in their NASH. And as you see on the left here, 43% of the patients getting selonsertib met their primary endpoint, and that was that over a period of 6 months, their fibrosis actually decreased. So there was actually regeneration of healthy liver. Compared that to controls of about 20%. So that's 20% is what you really expect to see in this population. And on the progression to cirrhosis side, it was very effective at reducing progression to cirrhosis. And if you look at the controls in green, 20% of patients, this is a 6-month trial, so this tells you how advanced they were. In 6 months, 20% of these patients progressed to cirrhosis. So this is a nice patient population from, if you're sitting in a pharmaceutical company, because now it's pretty easy to go to a payer and say, this is something you've got to treat now. It's not something that's going to take 20 years. These patients are at risk now. So when we first started, we weren't really quite sure how we were going to differentiate, but as you go through and do the drug development process, you learn. And we have found problems with selonsertib that frankly explains some of the reasons they've done things in their clinical trials and give us, again, have kind of given us the guideposts of where to go with our drug. So these are our crown jewels, and I can't share them except under NDA because nobody else knows about these. But they're life-threatening potential life-threatening safety liabilities. What's interesting here, if you go back to that last slide, 18 milligrams was the top dose. And that was the top dose in 3 different phase 2 trials. It worked in NASH. It failed in a diabetic nephropathy and a pulmonary artery hypertension trial. So wasn't enough to work on those drugs. We've gone back. It's clear that just wasn't enough for the drug. It turns out it was enough in NASH, but they couldn't give enough of their drug. They were clearly capped. So, and again, in phase 1, they went up to 100 milligrams. So they've got a great drug for NASH. If you could get a safer drug, you may be able to give more and get even better response in NASH, and then you can go into other diseases that are now completely inaccessible to that drug because they can't give enough. It also has some clinically relevant drug interactions, so I think in the NASH space, that's something that we can benefit from, and it really does create this opportunity for a superior ASK1 inhibitor. And we have that superior, we have several superior ASK1 inhibitors. So our first one is a liver-selective drug, and it's called SRT-015, seal Rock Therapeutics 15. So again, as we've identified what the problems were with that drug, we dialed them out through a lot of medicinal chemistry, but found ways to get around the issues that they have. And the thing that was a surprise, and a nice surprise, is that as we were going through and doing the pharmacokinetic work, we found it's got a 60-to-1 liver to plasma ratio. So now if I give the dose that I need to treat the liver, the plasma concentration is 1/60th of that. So that gives you an even greater safety margin, because in most tissues it's just not going to be present in enough levels to have any pharmacological effect. And then we have others that are non-liver selective that still have this benefit of not having the toxicities that, again, we're pointing toward some of the other diseases that Gilead has not been able to take their drug into. And again, what we're focusing on first is a particular kind of heart failure called HFpEF. So all of the work that I'm talking about and everything that we have done is our own. So like I said, we're virtual. We do everything through contract research organizations, nothing from any universities. We haven't had to in-license anything, so this is all our IP. We've filed 3 patents now. And frankly, our own IP firm, as well as several other from companies that are doing due diligence, have shown that there's no freedom to operate issues. So just a little bit of the data, and I won't go through too much of this, but first of all, just showing that the first order of business is develop a potent ASK1 inhibitor. And so this is just some data on ours in several different assays. So again, back to your pharmacology, an IC50 is the concentration it takes to get 50% reduction in the reaction. So low concentrations are good, so low numbers mean better activity. So, and just kind of, do we block the ASK1 kinase? Do we just shut it down? Our IC50 is at 17 nanomolars. Selonsertib is 7, and anytime you're in the low nanomolar range, it's fine. So we're good there. And then we have a couple of other assays where we actually take peripheral blood mononuclear cells. You stimulate them with LPS, and you can actually measure TNF release, and you can measure, again, activation of this P38 pathway. And again, we've got quite potent, much more so than selonsertib in P38 and around the same range in TNF. And then again, just finally showing that we also inhibit that junk pathway. So good potency there. And again, we've done a number of other safety studies, but I think the first in vivo studies that we've done to see, we've got all this great pharmacology, does it work when you put it in an animal? So the first study we did is an acute acetaminophen overdose. And so you give acetaminophen overdoses, you basically overwhelm the normal conjugation pathway through which acetaminophen is eliminated, and you get all these oxidative metabolites. And that's what causes the damage. And it turns out what's happening is those oxidative metabolites are going to the mitochondria, and that's stimulating ASK1, and that's what causes the apoptosis and so on in the inflammation in acetaminophen overdose. So this is a good model for looking at in vivo activity. of ASK1 inhibitors. So what you see, so this is the data from that study. So what you see on the left, the red bar is the data from the mice that just got high doses of acetaminophen with no active treatment, and you see ALT is the readout here. So 1700 or something on the ALT. And at again, so ours are the three bars on the left. So 0.1, 1, and 3 mg per kg, and selonsertib is the 0.3 and 1 in blue. So at 1 and at 1 and 10, let me think of the numbers here. At 1, we match. And again, we've got some explanations for why the lower doses, again, were lower, but again, the kind of concentration that you get in man is going to be much above of even the 3 mg per kg range. And down at the bottom is the gel, just showing that, again, the phosphorylation of P38 of junk matches that data. So again, encouraging data that said, okay, we have an active drug here that's working on the pathway. And again, this is the data that shows us that it's preferentially distributed to the liver. So in green is the liver concentration in mice that got 10 mg per kg of the drug. So you see that that's much higher. The blue is in the kidney, the red is in the pink, I guess, in the heart, and the red is in plasma. So again, this is the data that tells us we've got a very favorable profile for a liver drug. And we've given it multiple doses, and this doesn't change over time. So that's kind of it for the data. What I think is important, and actually luckily I think this fits with a lot of what Basel was saying, is we've spent a lot of time thinking about and starting to execute on our corporate strategy. So our goal has been do a deal basically M&A at the end of phase 1, because frankly with a clinically validated target, all you really have to show to a company is we have a good one, it's safe, it's got good PK, and we know the mechanism works. So you don't have to take this all the way to phase 2, phase 3. 2 years from now we should be done with phase 1, and as long as it works, you don't have to do any more. The rest, you do it there, and the rest is for them to do. So we've been engaged for 6-9 months now with pharmas. We've spoken to 20. There's a lot of interest in this target and what we're doing. Spoken to 20. 3 now are through we've had multiple confidential meetings through due diligence, and JP Morgan's this week, and we've got, frankly, one has said we've done everything we needed to get you a term sheet. The others are kind of about at that point. We've got the head of R&D of one of the big companies that's going to be there. So we're at the point where they're ready to move. What the deal structure that we're talking about here is, again, plays into Basel's discussion. So the primary route that we've been going is straight to these pharmas and saying, here's the proposition. We remain an independent company. We control all the development of our drug. You fund the company through the end of phase 1. Don't get any equity. That's just money to run the company with. At the end of phase 1, now, if you exercise the option and you want to take the product forward, there's an M&A at that point, and that's part of what we negotiate now. What is that M&A value? And then there's downstream milestone payments and so on. But the key here is it's non-dilutive funding, and we retain control. And the returns here could be substantial, because if you go back, there's been 5 or 6 phase 1 deals in NASH over the past several years. They've been upfronts between 100 and 400 million. And the full milestones are always $1 billion, but good upfronts, and especially if you're not splitting it up with the venture capitalists. So that's the way we're going. As a backup, of course, we're talking to the VCs. We're at about the same stage with them, but let's face it, the companies set a very high bar. So if you get something good from the companies, thanks VCs, but obviously if there's something, whatever, you have that as a backup strategy, but it would really have to be an amazing deal, or you'd have to just run in with the companies where they wouldn't be able to do it yet. So that's kind of the story. So we're right now raising $500,000 through a convertible note. And what this does is we're now in the middle of, I told you about the Tylenol model, which is not a fibrosis model. We've now started the run-in on a fibrosis model in the liver. And so we need to run that. Again, we'll do that versus selonsertib. We're doing that work now. we've kind of starting all of the things that you do, the toxicology, scaling up to kilogram quantities and so on. And in the meantime, so we need to keep that running. And then really this needs to go until we can get something, something signed with one of these pharmas. So that's what this really is. In a way, it's kind of a bridge financing. So our terms, again, they're on here, I can talk about them. They're kind of plain vanilla terms. We've raised $1 million so far with these, and Life Science Angels is a pretty tough negotiating group. So anything that's gone through Life Science Angels, you got to figure there's not much more for us to give. So anyways, I hope I've given you a sense of the opportunity here, how exciting the target is. I think we've got something very unique, and there's huge market opportunities. And then let's face it, there's a huge unmet need out there, and we hope that we can benefit those patients. And that's Seal Rock.
Mark, can we take one question? Pick out one question. We got to get on time. Any questions?
I don't see any.
Okay,
thanks.
Great. Okay. Neil, wait a minute.
Whoop. Oh, yes, the reason I came here.
That's what I was going to say. I'm going to waste 30 seconds at the most, but we couldn't have staged it better. I didn't know it was going to follow like this, but Basil, following your speech, that was one of the reasons, as my colleagues and I combed through 150 competitive companies for these awards we're going to invest in, that call it the DNA, Basil, I call it alignment of interest. We've got great alignment of interest, and pleased to present this to you,
Thank
Neil,
you very much.
and good luck to you.
Thank you.